as I know these fish aren't ultimately infected with gonorrhea. Okay, so my name is Suzanne McGaugh. I'm a relatively new faculty at University of Minnesota. And so as evolutionary biologists, many of us are interested in the evolution of traits. And a subset of us are interested in the genetic basis of those traits. And there's been a lot of discussion in the literature lately about whether or not we should even spend our time trying to track down the genetic basis. Because the idea is that many traits are caused by alleles of such small effect that even if we sequence every single individual in the entire species, we would never have the statistical power to track down those alleles, right? I'm going to argue, however, that in the cavefish system, that's not the case. And that's because these fish actually live in an environment that's so extreme and so different than the environment that their ancestors arrived from, that they are under really strong selection pressure. And that strong selection pressure, in combination with a lot of migration from the surface, creates a system where theoretically it's possible to get a genetic architecture that's based off of large effect alleles. Okay, so these fish are interesting for a variety of reasons. They lack pigmentation, you can see that. They lack eyes, you can also see that. They're also, if you look closely, they're really fat. So they have a totally different metabolism than the surface fish do. They have increased olfactory capabilities and increased taste buds, allowing them to compensate for the lack of eyes. They also have behavioral differences so that they don't school, they have lack of aggression compared to the surface fish. And one of the really cool phenotypes is that they have reduced sleep requirements. So if you look at this graph, the important thing to take away from it is that the black line is surface fish, and the red, yellow, and blue lines are cavefish of different origins, and total sleep is on the y-axis. So the big important thing to take away from this is that surface fish sleep a lot more than cavefish do. And this is the only fixed ecotypic difference I know of a sleep difference in nature. And so if you know of one, please let me know. But this provides us with a dramatically powerful system to map sleep requirements and the genetic basis of sleep. We have a genome, and it's an Illumina-based genome, but we are getting a PacBio-based genome. So that sets us up to do population genomics. And what we've done so far is we've sampled 3 origins of the cavefish, of the cave phenotype, and 2 surface fish origins, or 2 surface fish as well. We have Osteonyx aeneas, so the Osteonyx mexicanus is the cavefish. Osteonyx aeneas is a congener, and what that allows us to do is to polarize genetic changes to say this evolved in the cave and it's derived in the cave. Okay, so we have 43 samples. After we've aligned them to the genome, we're left with actually pretty decent coverage. And what we've done is we've started with really large effective things that we think would be really large effect compared to non-synodic or just plain old non-synodic changes, right? So we've put all of this data through variant effect predictor through Ensemble. And what that gives you then is splice donor acceptance variants, splice region variants, initiation codon polymorphisms, these things that should cause really large phenotypic effects. And what I'm going to tell you about for the rest of the talk are 2 different examples of stop codon polymorphisms we think cause phenotypic changes. So these are premature termination codons. So what we do is we use that congener and we use the surface and we say, okay, they have the long form. Now we're going to look at the caves, and we've got these premature termination codons in the caves. We selected for things that were only high frequency in the caves, low frequency in surface, and never present as homozygotes in the surface, so that we're thinking these should cause major phenotypic changes in the caves. This is done by an undergraduate student and a tech. Okay, so one of the ones we found was fixed in Molino Cave. This is a gene that underlies Hermansky-Pudlak syndrome number 4. It causes about a 70 amino acid truncation. You can see that offspring that have this syndrome in humans, they are albinos, and they have ocular problems as well. One thing to note, this is also under an IQTL, eye size QTL in the cavefish. The next thing I want to mention is a sleep gene. This is PER3, period 3. You might have heard this in the news that it underlies being a LARC phenotype in humans. And so the important thing to note here, this is a knockout mutant of mice. It's a homozygote knockout. We have wild type and then the knockout mutant. So you can notice that the PER3 mutants sleep a lot less, and that is it. Oh, we're doing CRISPR on these guys. Thanks.